in a meal. When I was your age, I had nothing, stand between me and it. So I'll move through this as quickly as comprehension allows. I want to talk about a completely different set of projects we worked on in my lab from what I spoke about in my last talk. And my title involves the mitochondrial life cycle of neurons, but the kind of overarching theme of this study is in line with some other things we've done in the lab now for going on 20 years. And that is addressing the old idea that comes out of the 60s and 70s, that the axon is more or less a kind of inert tube, along whose surface action potentials are conducted, and up and down the length of which material is moved at its origin entirely in the cell body or entirely at the growth corner site. We've come to revise our views about this in recent years. For instance, some things are undisputed. It's been clear for 30 years that there's abundant lipid synthesis in the axon. It's clear now that there's protein synthesis, although the amount and its significance remain extant research issue. What I want to talk about is the notion of whether some important aspects of the mitochondrial life cycle, above and beyond their movement up and down the axon, can occur in the distal axon far from the cell bodies. I mentioned in my previous talk that there's kind of three categories of questions that exercise my lab about mitochondria. The first is their movement and distribution. How do you get them to the right place at the right time in this enormous engineering project of the cell? And that's mostly what occupied my previous talk. I want to talk about two other aspects of the mitochondrial life cycle this morning. One is their metabolic activity. In particular, is it modulated somehow to serve the needs of time and place? Again, in this extraordinarily large and asymmetric cell. And that's an issue that I've been very interested in for a long time, although we've made less progress than I would like. And that is their proliferation and biogenesis. And that is, do mitochondria do some of the things that they do to reproduce themselves, make new stuff, replicate their DNA, grow, divide, fuse with each other, out in the distal axon? Or do they only do it in the cell body and then visit the axon? And thinking as a cell biologist, this is a profound question to me, because it means the mitochondria in the distal axons that innervate the muscles of your foot, which are a meter, as we've discussed, from the cell bodies. Are they just visitors? Did they come all the way out from your lumbar spine to distal axons in your foot, which probably means they barely have time to reside there before they turn over? Although that's a possibility. That's one view, that they're visitors out there. If, in fact, they can undergo significant parts of their biogenesis in the axon, that's a completely different view. That implies the possibility that the mitochondria out in that distal axon in your foot have been in the distal axons of that nerve since you were in utero, and that they are the descendants, if you will, of ancestral mitochondria that have never visited the cell body. Okay, and again, why does this matter? Because we're talking about this absurd dreamlike cell that has very different needs for everything, including mitochondrial function in a region like this, the somatic dendritic compartment, a region like this, the nerve terminal, and a variety of regions along the axon, which in many neurons occupies the vast majority of the biomass. So, how can we modulate how much mitochondrial function is in different parts of this enormous cell? Well, the obvious way that's been a theme of this meeting so far, and that I talked about previously, is by regulating transport. And you were talking about 1993, Peter. In 1993, Bob Morris and I published the first evidence that when a region like a growth cone in a growing neuron requires additional mitochondrial function, that regulated transport delivers and deposits them there. That's a no-brainer at this point in the course of our thinking about axonal transport. But there are other possibilities, and one that we've thought about for years and tested in a couple of ways that I'll show today is, what if in a region where you need additional mitochondrial capacity, ATP production, calcium sequestration, and so on, you could locally regulate their metabolic state and change the activity of electron transport, ATP synthesis, and so on. And then the third possibility, the one I was just alluding to, probably the weirdest and most heretical, what if they could proliferate locally at a great distance from the cell body. That would also supply additional mass of mitochondria and presumably additional function. So first I want to talk about the possibility that you have local regulation of their metabolic activity. And thinking about this arose from observations in a study I thumbnailed before for you, and I'll thumbnail again. It's not what I want to talk about, but it was the leaping off point. Sunita Chadha and Mahabaleshwar did experiments a number of years ago in which she applied polystyrene beads to which had been covalently coupled growth factors and other molecules, put them in contact with axons, and showed that she could induce local docking of mitochondria. Now, I did mention this before. She could cause them to halt at sites of nerve growth factor stimulation, nerve growth factor being an attractive cue when these axons are growing out, and in regions of semaphore 3A stimulation, this being a repulsive factor. And I believe I mentioned before that this local docking required both the actin cytoskeleton and PI3 kinase signaling downstream from receptor tyrosine kinases. That's not what I wanted to tell you. The reason I wanted to tell you is that something she sort of noticed in passing is that the mitochondria docked in this region, when she used voltage-sensitive dyes, seemed to be brighter than mitochondria in the axon as a whole. So this is a kind of a chance observation, but it made us wonder, is local signaling not only regulating their movement and distribution, but possibly locally regulating their metabolic state? And so let me show you some of the experiments we did to look at that. So the hypothesis would be, is there something going on in neurons that can locally up or down regulate the mitochondrial metabolic state? We did these experiments in DRG neurons, and I dug back into the vault, sort of in the basement vault, to show you this picture of a very early DRG neuron. This is a chick neuron that's been set out just for 2 hours in culture. It's got an unusually pretty growth cone, which is why we took a picture of it. A short but rapidly growing axon in the cell body. I dug this out because I knew I'd be talking to young people. And these things over here, those are scratches on the film. This is a picture taken with film, and if you're curious about photographic film, I can tell you about it at lunch. Okay, so what do we do with these things? We use tetramethylrhodamine methyl ester, which I described previously, a voltage-sensitive dye in the sense that it's a lipophilic fluorescent cation, so it partitions to the negative side of the membrane with a potential across it. It's non-toxic, equilibrates very quickly. And what we did is to take the fluorescence of the mitochondria, divided by the background fluorescence of adjacent cytoplasm, in essentially a Nernstian treatment, so that we know the dye ratio from the mitochondria to the cytoplasm has a logarithmic relationship to the intermitochondrial membrane potential. And we asked a couple of basic questions. One was, if we look at different regions of a sensory neuron, again, growing in a dish, not in an intact nervous system, do we see different metabolic states as evidenced by different intermitochondrial membrane potentials? And we looked very hard at this because we had a couple of suspicions. We wondered if mitochondria in the growth cone region, where we presume on the basis of biochemical studies you have a greater need for ATP, might be in a higher metabolic state. We wondered about branch points because in neurons in culture, as I guess you could argue there's evidence from neurons in vivo, you have an accumulation of organelles, including mitochondria and branch points. So a lot of stationary mitochondria. We're very curious about that. And then just generic vanilla sort of regions of the axon. Again, in a neuron growing in a dish without complex physiological memoirs, but with a uniform environment. And what we found, to our disappointment, is that the membrane potential is extremely similar across all sort of places in these neurons. Axons, branch points, pretty much the same. The one thing we did see is that in the immediate region, within 10 or 20 microns of active growth cones, the dye fluorescence ratio was up by almost 1/3. So nothing dramatic as far as regions where they're stationary. Another issue we addressed that's been a little bit contentious. Oh, well, let me make one other point in case you ever use a technique like this. Something that's often not appreciated about membrane potential in live cells is that it's actually variable from cell to cell. And this is something that no sort of mitochondrial biophysicist has been able to explain to me. And I've talked to these guys a lot. So this is sort of inside baseball, kind of popping the hood. Data at this level of details, not intrinsically interesting for this plot. I just want to make the point. If we look at a number of individual cells, and we plot the median plus or minus percentiles in whisker plots here, you get a sense of the variation of the measured mitochondrial membrane potential of all the mitochondria in a cell. What we see is that the spreads in individual cells are actually not that great. So this is the fluorescence ratio for 11 different cells. But the variation among cells is pretty big. And again, this is even more sort of pin-headed detail. Let me just make the point that the standard deviation of the means of the mitochondrial dye ratio for each cell is quite a bit bigger than the mean and standard deviations for each cell. So if you should read a paper that uses a technique like this, or get one to review as you get a little older, this is a question you want to ask yourself. All right, in any case, we use these comparisons amongst different mitochondria of the dye ratio to ask a couple of different questions. Probably the most important one was, there's been an issue of whether mitochondria that are outbound in the axon, anterograde moving population, are somehow more robust, younger, more robust metabolic than the ones coming back. And you heard an exchange between Professor Saxton and me, I think, around the time of my previous talk, in which he insisted, I believe, that I'd be less polite about this issue. But, and although I'm from Chicago, I'm always polite. So let me just say that despite the data in the literature that indicates this is likely, we found, in fact, a very careful observation, no differences in mitochondrial membrane potential between anterogradely moving and stationary mitochondria within the same axons, or between retrogradely moving and stationary mitochondria, or the coup de grace. We found no difference in membrane potential in anterograde and retrograde mitochondria. I consider this issue resolved the best that I can, although an important caveat here again is the easy place to do this is with a neuron in a dish. A neuron in a dish is an embryonic regenerating neuron. It's not very old, and it has a physiologically uniform environment in the culture. This is something we're about to repeat to the best of our ability in the intact fly-larval nervous system that I talked about and that Dr. Saxon talked about previously. So, no, we can't distinguish differences in metabolic rate using the standard membrane potential between anterograde and retrograde mitochondria. Okay, what else? Are there other experimental variables that we could use to induce these changes? Well, there were, and this circles back around to the issue I raised to kind of kick us into this, of local stimulation of cell signaling. So again, these are these experiments where you covalently couple something of interest, a growth factor, a repulsion factor for axonal guidance, to a polystyrene bead. You put them in the culture, they contact axons, and then you ask what's happening to the mitochondria in this region. We know they accumulate there, and so what we did was to look quantitatively at the membrane potential. And in fact, it does go up in the region of either attractive or repulsive signaling. And so what we did was to look at the red bar is the membrane potential ratio on the y-axis in the immediate region of the bead, that is the 10 micron stretch of axon immediately adjacent to the bead. The next bar, you probably can't read this font from the back, immediately next to the red one was the next 40 microns on either side. And then the leftmost bar for each set here is farther downstream in the axon. So whether we looked at the fluorescence ratio in the neighborhood of an attractive factor like MGF or repulsive factor like semaphore, it went up about 50% from the general axon to the immediate region of that bead. A boiled control bead produced no effect at all. Now, not only was this stimulated by these cell signaling events, but in trying to do some pharmacological studies, some inhibition studies, we happened to put onto these cells, not with beads present, but just in a dish where we could make voltage measurements with this dye, a couple of inhibitors of receptor tyrosine kinases. And what we found, this number here is the data that came from Boston, which is a receptor tyrosine kinase inhibitor. The application of that in the bath to these cells decreased the dye ratio, this measure of voltage, by 75%. And I point out, as I did in a different context in my previous talk, you have to apply to neurons in the dish not just mitochondrial poison, but more than one category of mitochondrial poison to diminish the membrane potential by that much. So there's something profound going on with signaling downstream from receptor tyrosine kinases. kind of disappointing in a sense that very few people are working on the issue of the interaction between mitochondria with this sort of visitor that came to stay, and the rest of the cell signaling that's going on. A possible exception, I guess, being Stefan Spratt at the University of Iowa, whose work I heartily encourage you to read if you're interested in that. So where this hangs is, there's clearly evidence that one can experimentally manipulate the metabolic state of mitochondria and neurons. There's one little nugget of physiological difference, the region of active growth, but no evidence we can find that it varies with direction. Yes?
When you say, so you're saying mitochondrial activity, right? So when you say mitochondrial activity, it means that it is what?
I say mitochondrial activity to be deliberately vague because we can't directly measure ATP synthesis. We can't directly measure yet, although we're working on something like oxygen consumption. We're using the membrane potential represented by the dye ratio as our stand-in for metabolic state. It isn't like we can say this dye ratio is metabolic state 4, this is metabolic state 3.
Yeah, so other thing is, so increasing or decreasing dye uptake is also depend on calcium in the mitochondria. It can polarize based on high level of calcium. So
higher low calcium. Calcium won't, I mean, calcium uptake probably won't affect the dye ratio. But the important thing to remember about mitochondria is virtually everything that they do. The import of proteins, the generation of ATP, the transport into the matrix of calcium, they all depend on the intermembrane potential. And in fact, something interesting that the mitochondrial biophysicists have urged me to be aware of is that when you do partially uncouple mitochondria, when you do apply modest amounts, modest levels of mitochondrial poisons, mitochondria continue to produce ATP to the bitter end, and it's the other functions that seem to get sacrificed. Does that answer your question? All right, so the second issue I was talking about, the local proliferation of mitochondria. Do we have evidence for that? And it's actually really worth asking first, what do we mean by mitochondrial proliferation or biogenesis? I mean, what are the things one could measure that are involved? These aren't cells. They used to be 2 billion years ago, the respiratory bacteria, but they're now stuck with us and we're stuck with them, and it's a pretty good relationship. What is it that they have to do so that we have enough of them, so that they double, for instance, before a cell dies? Really important thing to understand, we tend to look at mitochondria either in fixed images with some kind of a fixable vital dye, or in live cells with the vital dye, or with an antibody stain or something. We look at them frozen in a moment in time, or we look at them with a particular time lapse, protocol for movies. And I think it creates an illusion that mitochondria exist for long periods of time in those cells as individual organelles with their own identity. In fact, if you look with the right sort of cell-relevant time sequence in a movie, for instance, at a macrophage, a fibroblast, and epithelial cell in culture, what you realize is that mitochondria are normally a kind of syncytium. There are even places like the somatic dendritic compartment of hippocampal cells where the mitochondria are pretty much all connected to each other. And what happens is out of this sort of Borg collective-like mass, off-bud individual mitochondria, and they exist individually for a while, minutes in some cells, hours in others, maybe days in some neurons. But they fuse with each other all with the continuum. And so there's kind of the illusion of individual organelles. The same way if you were focusing on Golgi vesicles, you might miss the point that the Golgi is a unitary organelle with tons of budding and fusion. Okay, now the axon's a special case. Mitochondria have to be fairly short on the scale of things to undergo efficient transport. And so we assume that in the axon they have an independent identity as independent organelles that lasts longer than it does in most other times and places. We think. One possibility that's proven maddeningly hard to evaluate is the idea that there's a kiss-and-run phenomenon going on. You've probably heard this with respect to exocytosis, but David Chan has amassed evidence in non-normal cells that mitochondria just en passant can contact each other, and believe it or not, fuse both their outer and inner membranes long enough to exchange contents and divide from each other again, all in a matter of seconds. If that's happening in the axon, we're sort of screwed because it would be very hard to detect. It's something we're thinking about. All right, so we've tried to observe and quantify, well, we have observed and quantified 3 things. directly around it. One is mitochondrial DNA synthesis. Don't forget, they really were respiratory bacteria living free only 2 billion years ago. And although they've coughed up most of their genetic content to the nuclear genome, they had this little circular genome. And in us, in humans, it has 13 proteins encoded. Once they're too greasy to import. And it has RNAs. And it's got to replicate that. The other thing is mitochondrial fission, because of course when they grow and divide, and mitochondrial fusion, the function that makes them part of this sort of Borg collective. All right, so how do we do that? To measure mitochondrial DNA replication is difficult, and in the past, I think conclusions have been drawn from studies kind of in the late 90s and before, based upon negative evidence coming from techniques that were between 10 to the 4th and 10 to the 6th, less sensitive than are available now. So this is an image from Madonna Mary's work in the lab. This is a sympathetic neuron, actually, from a chicken culture. This is a mitotracker staining this fixed cell, and this is BRDU labeling from a pulse of several hours of BRDU to try to detect mitochondrial DNA replication. Now, the reason these labeled mitochondrial genomes are so large and blotchy is that in order to detect the replication of this tiny little genome, we needed to use a double precipitation technique, which Madonna developed. But it increased our sensitivity enormously and allowed the studies of the spectrum the next few years to be done. Now, does detecting a mitochondrion whose DNA has been replicated during the 4 hours or whatever that you pulse the cells with BRDU show that there's mitochondrial DNA replication in the axon? Of course not. The DNA of this mitochondrion could have been replicated in the cell body, and then 20 minutes before we fix the cells, it could have moved out into the axon by axonal transcript. So an image like this tells us we can detect mitochondrial genome replication. It doesn't tell us anything yet about whether that replication can occur out in the distal axon. Okay. All right, so how do you figure out whether mitochondrial DNA replication, once you can label it, does occur in the distal axon? Well, you have to do an experiment that I'm glad I had a younger person with steadier hands to do, and that is the following. And it's an experiment we've done in the past to look at protein synthesis. That is, you can set out, not a cell, but this is a ganglion, a sympathetic ganglion from the chick. It contains something on the order of 10,000 cell bodies. You set it out in the dish, you grow it for 4 or 5 days with mitotic poisons so that the non-normal cells don't proliferate, and you end up with a halo of axons radiating out. And this is shown as little gray lines in tens, but in fact there's hundreds to thousands of axons. And we can get this out to about 5 millimeters in diameter, 5,000 microns, or a little more. So what she did was to exotomize, but the point here is we're going to remove the cell bodies and leave behind the axons. And they'll do fine for 8, 10, 12 hours. And so if you remove the cell bodies and you have just, from the mitochondrial point of view, mitochondria and axons with no communication with the cell body, and you then label with BRDU, you should be able to determine whether you in fact get mitochondrial DNA replication out in the axon at a great distance from the cell body. So this is a high-mag view of one of these ganglions. Here's the ganglion view like this. Here's axons and fascicles of axons, and this shows the exotomized version with the cell body mass gone. Okay, so what she did then was to do the exotomy, remove the cell body mass, and then pulse the cell body-free axons with BRDU and look for indications of mitochondrial genome replication. And this shows mitotracker in this panel, the BRDU stain after fixation here. And here are just 4 examples of mitochondria whose genomes were replicated during the period of BRDU labeling. So what did that amount to? Well, we wondered how frequent it was, and so a kind of experiment that we carried out a number of times was to quantify the frequency of recently replicated mitochondrial genomes in axons that were attached to cell bodies versus ones that were not attached to cell bodies. And I'm actually going to show you one more time here data that's a little more detailed than necessary, again, to make a point about this kind of experiment. So what you see here is the percent of mitochondria that have replicated DNA, replicated during the BRDU pulse on the y-axis, and a couple experiments here. You see the values for an intact ganglion, axons in an intact ganglion, versus mitochondria and axons with the cell body mass removed. And I'm showing you this to make the point that we don't have a good grip, I think, on the physiological parameters that make mitochondrial replication frequent or infrequent normally. And so this is 2 representative experiments, and you see we have quite a bit of variation in factor of 2 between the percent of mitochondria replicating their genomes per unit of time, just in the intact neurons. But what we found is that the fraction of DNA synthesis in mitochondria that we got from exotomized cultures was pretty well clustered around 1/3 as much hypertrophic DNA synthesis. So again, this means you look out in the axon during a given period of time, a time when little label, and you see mitochondrial DNA replication. About 1/3 of that occurred in the axons rather than in the cell bodies, followed by conveyance out. Yeah.
So what would be the effect of those local signaling that you talked about on mitochondrial replication?
Oh, good idea. Would you like to do that experiment? Because I tried to find someone who'd like to do it for 4 years and it didn't work. Yeah, it's a hard experiment, but it's a very good idea. It is.
And for how long you are giving a BRD pulse?
The BRD pulse in the experiments I just showed was 6 hours.
6 hours.
Yeah. The axons remain in pretty good shape for up to about 12 hours after we remove the cell body masses.
Does that change the ratio if you just check after 6 hours or 12 hours?
Well, sure. And in fact, I don't have this data here slide with me, but we were obviously interested in the rate of turnover of the mitochondrial DNA. And so we did kinetics. We did a variety of lengths of time, 1 hour labeling, 2 hour, 4 or 6 hours, and determine a T1/2. I can't remember off the top of my head what it was. But clearly, the longer you incubate, the higher percentage you see.
Yeah.
Does that answer your question? Are we good? Okay. All right, so what about other aspects of biogenesis and whether they occur in the axon? We were interested in fission and fusion and whether they occur at a distance from the cell body or whether they purely occur there. I've been looking at mitochondria in live axons for so long that I have hundreds of hours of records on what's called videotape. And again, if any of you young people are interested in videotape, I'll explain what it is over lunch. And we certainly have examples where we think we're seeing fission. In other words, we see something moving that looks like a single mitochondria, and then it appears to become 2, and off they go. We also have examples of what looks like 2 mitochondria turning into 1 and moving. But I haven't really been able to convince critical observers, which is to say reviewers and study sections, that these don't constitute coincidental movements of, let's say, 2 mitochondria that were traveling in train and then separate, or 2 mitochondria that were moving separately and then traveling in train. So it's been a very difficult issue to work on, and it's being pursued in both my lab and Dr. Sax's so far without definitive results. So what we asked at this point was whether other approaches could give us strong or indirect evidence for fission and fusion. And what Madonna and I did was the following. We focused on the fission and fusion proteins and wondered whether we could visualize them and under or overexpress them and get an idea of whether mitochondrial fission and fusion might occur out in the axon at the great distance. Just by way of review, I think we've heard about these already, but the major protein driving mitochondrial fission is DRP1. It's a dynamic-related protein. The energy provided for its function presumably comes from GTP hydrolysis. And it's shown as it is here because there is a soluble, I use that tricky word, there's a soluble pool of DRP1 that is not associated with mitochondria all the time. And the belief from studies in non-neuronal cells is that it is recruited to mitochondria fairly shortly before a fission event, that it drives fission, and then pretty soon after the fission event, you would find it dispersed again into the cytoplasm. So in an incipient fission, you'd expect to see it near the center of the mitochondria. If you're capturing a view of a mitochondria shortly after fission, you ought to see smaller mitochondria and DRP1 at the end. Okay, how about fusion? This too we've heard a little bit about already in this meeting. Oh yeah.
So, like, how like
How what?
How it applies to constitutive force?
I don't think that's perfectly clear yet. I mean, it's a dynamic-related protein. I don't think the mechanism is identical to the dynamic. Although the notion that you form a ring that can exercise a pinching force makes me presume that it would have a similar mechanism as a dynamic. Is it a conformational change driven by GTP binding hydrolysis? Does that make sense? All right, so in the case of fusion, there are two key proteins. One is mitofusin. There are two in most vertebrates. And this is the protein. You see two of them undergoing a homotypic interaction here that drive the fusion of the outer mitochondrial membrane, and OPO1, which we heard about previously at this meeting, drive the fusion of the inner membrane. So we focused on DRP1 and mitofusin for these experiments I'll describe next. The first thing was just that we just stained mitochondria and stained DRP1 on the axons, and we found something that very much surprised us. Now let's think about anticipated results. If mitochondrial fission occurs only in the cell body as part of a biogenesis that's restricted to the cell body, and then mitochondria enter the axon, you might still expect to find some of this fission protein on mitochondria, which had, just very recently divided in the cell body and hadn't thrown off the DRP1. If that were the case, the farther you get from the cell body, the less likely it would be that a mitochondria you visualize had DRP1 on it. But in fact, when we looked at mitochondria, this is a fixable mitotractor stain, and we looked at DRP1, the green stain here, and we looked at the fusion image, and we looked at mitochondria, and we scored as we walk away from the cell body into different zones of its length, what percent of mitochondria, in fact, have a DRP1 signal. It didn't vary much at all. And so this is the proximal 100 microns of each axon. This is the distal most 100 microns, and this is the middle 500 to 800 microns of the axon. You see the percent of the mitochondria that were DRP positive dipped a little and came back up at the end. So no evidence that it's falling off the distance. Furthermore, and this is something that Madonna had a hunch she was seeing, and so we went ahead and quantified. If we looked at mitochondria, I hope you can see this image from the back, that have DRP1 at the end versus ones that have DRP1 in the middle. If this represents DRP1 that's sitting on the mitochondria about to immediately incipitly divide it, or just after a recent division, then these ones with it at the end ought to be a lot shorter than these, and in fact the lengths differ by more than a factor of 2. So what we were seeing is not only lots of mitochondria, more than 80% of the mitochondria in these growing axons, with this fission protein on their surface, but it was distributed in a fashion that strongly suggested that fission was occurring, consistent with fission in the axon. I'm sorry?
The green is DRP1.
The green is DRP1, the red is mitochondrial.
It doesn't enter into the axon. The DRP protein.
Well, yes, it's on mitochondria. So
the scientific portion of the axon.
Don't know. I don't know. So is DRP1 part of, say, a slow transport component? I don't know, and I'm not sure whether that's personal ignorance or universal ignorance. Don't know whether that's known. But in any event, we find it on most of the mitochondria in the axon. Okay, so mitochondria way out in the distal axon, loaded with this fission protein. It's distributed in a way that suggests fission events about to happen or fission events that have recently happened based on the length distribution. And so we tried another approach to this, and that was to knock down DRP1 with the hairpin RNA transfected into these neurons and thereby suppress mitochondrial fission. And when we suppressed fission, we found, well, here's the control cell. You see the mitochondria, although not real on this slide. Here are some examples of the proximal axon near the cell body, the middle axon, several hundred microns from the cell body, and a distal axon, many hundreds of microns from the cell body, in cells in which we expressed a hairpin that knocks down DRP1. And what we found, and it's not wonderful lightness here, but we found much longer mitochondria. And so let me show you the length distribution. If we looked at the length distribution of mitochondria in the control cells, you see it here. It's one-tailed, the way we've broken out the histogram here, with a mean length around 2 microns. Enormous difference of more than a factor of 2 when we inhibited mitochondrial fission by knocking down DRP1. What does this mean? We have very long mitochondria out of the distal axon far from the cell body, including this half of this distribution. Mitochondria so long that they can't undergo axonal transport. Mitochondria up to about 8 microns show no relationship between length and motility behavior, but when they get up to 10, 12 microns or above, they simply can't move. So we stored hundreds of mitochondria in these DRP1 knockdown cells, way the heck out in the distal axon, and they're not capable of moving. That tells us they must have arisen in the distal axon, which tells us indirectly but quantitatively that mitochondrial fission occurs in the distal axon. Now we came at it from the other direction, and that is to try to stimulate fusion, so we overexpressed moderately mitofusin 1 in these cells, one of the two mitofusins that they have endogenously. And we got a very similar result. That is, control cells, the typical short mitochondria, the overexpression cells, you can see the mitofusin on them, and you see much longer mitochondria. And let me show the quantification of that as well. So, overexpression of mitofusin, the primary fusion protein, longer mitochondria. Here again is the control distribution in black of mitochondrial length, a frequency distribution. And here the distribution in the case of mitofusin 1 GFP overexpression. Once again, more than a twofold increase in the length of the mitochondria in the axon, including large numbers of mitochondria of a length in excess of what can move at all at an enormous distance from the cell body. So again, I guess I would contend quantitative, if indirect, evidence that fission and fusion are occurring very far from the cell body in these axons. Okay, so what's going on in the axon? What am I trying to convince you of here as far as the mitochondrial life cycle? And again, let's look at this through the prism of, are these just visitors in the axon, or can they actually undergo various parts of their life cycle, which include things like DNA synthesis and regulated phenomena like fission and fusion at a great distance from the cell bodies. Okay, so motility and docking can be controlled by cell signaling, but it remains sort of early days in understanding what's going on there. When that does occur, find membrane potential can vary regionally, as we see it vary in growth cones, at least in isolated cells in a dish, but probably does not vary between anterograde, retrograde, and stationary mitochondria. And if Dr. Saxton was here, he would say, stop saying probably. It just doesn't vary between anterograde and retrograde. Okay, what else? Membrane potential, to our surprise, that is intermitochondrial membrane potential, responds quite powerfully, not only to attractive and repulsive signals, but to RTK signal, you recall it, we were able to depress the membrane potential as much by inhibiting RTK signaling as we could with multiple mitochondrial poisons. So I think it's clear they can replicate their DNA within the axon, that studies from 15 or more years ago that implied this was not the case suffer from really inadequate sensitivity. And in fact, that the amount of DNA replication, mitochondrial DNA replication that we can visualize in the axon represents a very large fraction, perhaps a third of the total neuronal mitochondrial DNA synthesis. And this could be considerably larger in neurons in which the axon represents a larger part of the total biomass. Fission and fusion almost certainly occur in the axon. I'm saying almost because we made indirect measures, including very far from the cell body, and I do hope that we all see direct evidence for this before too long. Okay, the caveats to this is not done in the intact nervous system. It's done in cells living in a dish in a uniform physiological environment without landmarks like synapses, without landmarks like nodes of Ranvier in myelin cultures. All right, so that's what I wanted to tell you. Let me recognize the people who did this particular chunk of work. Jessica Berber, who did the potential measurements in DRG neurons, and Donna Amir, who carried out the fission fusion studies in sympathetic neurons. And since I'm up here giving my last talk of this conference, I'd like to thank you all quite sincerely. First, to thank you for the invitation, which was generous and has led to a wonderful first experience with this country. And to thank all of you for these excellent scientific discussions. It's been a very stimulating, somewhat jet-lagged week plus. And finally, just to thank you all for your kindness, which has made this week really a pleasure. And so thanks for your attention. We'll get to lunch in a timely fashion, but if you have any questions, fire away.
Yeah, we still can have some questions.
Yes.
Like in neurodegeneration, it has been observed that there is large need of NGF, the nerve growth factor, from the below. So there is some study done with mitochondrial potential and NGF in that. Because in that way, the enhanced it affects it causing overexpression of many cancers.
I don't understand the question.
In neuropathic patient, there is enhanced NGF that we told about NGF and mitochondrial.
Right.
So in that way, there is enhanced. So if you have to study that, effect of NGF neuropathic on mitochondrial.
Not that I know of. No, I'm not aware of such a study.
Can you repeat the question? I also couldn't get it.
Could you repeat the question?
This thing, so there's so much echo that it.
Actually, what I did is neuropathic pain.
Neuropathic pain, okay, which I'm an expert on, by the way. I have no L4, L5. Yes.
There is the axons, nerve links are separated from the site where they are attached. So this gives in this time to enhance pain, insensitization.
Right.
So one of the major factors in insensitization is nerve growth factor.
So nerve growth factors are the major factor
one
in
of the major
sensitization.
factors. So there is some study I wanted to do related to mitochondria and NGF.
No, I mean, to my knowledge, the two things we've published are it. I mean, I assume when we publish Sunita Chadha's work and then Mandana's work that someone in the signaling field would jump on this with both feet, because frankly, I'd rather have someone else do it than us. But it hasn't happened. So you study neuropathic pain?
Yeah.
Oh, we'll have to have a talk, because I experience neuropathic pain. Yeah.
So actually, so there was one point, especially even with the regular mitochondria have no potential project, it doesn't actually kind of judge on that. So do we know that the mitochondria is too retrograde? Will that end up in cell body, or is just petrol around the
So his question is, okay, you shot down this idea that the outbound ones have high potential and the inbound ones have low potential. Is there evidence that the ones coming retrogradely are destined for the cell body and or destined for turnover degradation in the cell body? This is a tricky question. People often cite EM studies in which a block of some kind allowed mitochondria to build up at the proximal and distal sides. And the ones on the distal side, which are becoming retrogradely, look sort of bad, they look senescent. But if you read all the studies that have been done in that area, there's also studies in which they look about the same on both sides, and even one study in which they look worse on the proximal side. And so I think the issue of where most mitochondrial turnover occurs remains open, because mitophagy is kind of a space-consuming process. I think of them as getting back to the cell body to be engulfed. But now we have recent evidence from Erica Holzbauer's lab, at least in the cultured neuron, that you have co-localization of autophagic vacuole proteins and mitochondria way out in the distal axon. We haven't seen that in the intact nervous system yet of the flock, but we've only just started looking.
Yes, the question was not in terms of mitochondria, but there is when mitochondria move into the axon. I mean, into the body from the axon.
Do they move into the, yes, some mitochondria do move into the cell body from the axon. We know that just from having made observations in the village. In other words, they don't come all the way back and then chill out for a while and then go back out again. Don't see that. You rarely see a persistent change in direction of the anterograde or retrograde populations, even though on a short time scale they can undergo wrong direction. It's just a
specifically photoactivate, just I'm carrying over from that. If you specifically photoactivate the mitochondria near the synapse, and if you monitor that, it would be majorly retrograde, right?
Oh, yes. Yes, I think we haven't been able to do this yet, although Bill Saxon was built to fly. We don't have it from there. If you did photoactivate the neuromuscular junction, I expect a lot of them to stay put for a long time, and some subset to get onto the conveyor and undergo retrograde transport. Peter.
I was very interested to hear you speak on the relationship between the length of the mitochondria and whether it's mobile or stationary, because the numbers you gave are actually remarkably similar to microtubulars, that once the mitochondria got beyond, say, 10 microns, that it was stationary. And I think I've heard Tony Robbins say similar things about mitochondria, that really there's beyond a certain length, they just don't do. And I'm wondering if you pondered why that might be.
Sure, I've not really pondered it. I've discussed it at length with Tony Brown. So Peter's question was, gee, your numbers for how long the mitochondria has to be, sort of 10, 12 microns, before it just won't move, are awfully similar to the numbers for how long neurofilament or microtubule has to be before it just won't move. And in fact, I've talked about this at length with Tony Brown, and we don't know what it means. I assume that it has something to do with Reynolds number issues and viscosity, but it may not. It may be that above a certain size, there's a regulatory event that persistently docks mitochondria. Now, it's important to keep in mind, under normal physiological conditions, you don't get very many really long mitochondria. You tend to have a mean in the cells that I was showing here, the cultured DRG neurons, of a couple of microns and not a big sigma around that. So, suppose you had a question.
I thought it was pretty interesting that almost every mitochondria in the axon that you looked at had DRP.
Yeah, it's over 80%. Total surprise to us.
And I think your explanation is that I understand that you want to promote the idea that.
No, I'm not promoting anything. I'm following the data.
Promote is not very good, but put forth the idea that there's fission going on. Obviously, it's going on in the axon. But I was more thinking that if you really have DRP on every single mitochondria, I wonder if there's a second order regulation of DRP that we don't understand.
Oh, good point. Good point. So he's asking, what's the deal with all this DRP and mitochondria? And you asked a question whether DRP is transported into the axon. Maybe it's mainly transported into the axon on the back of mitochondria as a mechanism to allow mitochondria to undergo fission while in this distal axon. But in fact, it's not active until, as you described it, some secondary regulatory.
So we don't understand anything.
Yeah, that's actually a very appealing idea to me because it didn't make sense to us from the beginning, and we reviewed it very carefully, that 85% of the mitochondria in the axon, where a significant but modest amount of fission occurs, should have the fission machine.
So there's something else going on.
Yeah, I think a secondary regulatory event that's kind of axon specific is a very cool idea.
In cell body, mitochondria don't have DRP, so much of DRP.
Sorry?
Compared to the axon.
Yeah, well, we can't, it's really hard to see anything in these cell bodies, but if you go to a non-normal cell, it's much more. You see it on, you tend to see it on mitochondria just before fission or just after fission, and then off it goes, dispersing into the cell.
In the picture, there was a lot of DRP in the cell.
Yeah, there's a ton of mitochondria there, and there's also soluble DRP. So the cell body is just a diffuse glow.
Yeah, so is there any bias of fission events towards anterograde or retrograde movement?
Is there any bias of fission events for anterograde versus retrograde? I can't say because our observations are being made in fixed cells. And so when we see a mitochondria that's short and has DRP at one end, we have no idea whether it was stationary, anterogradely, or retrogradely moving at the time. I will say this, when we in an unrelated study, knocked down myosin 5 neurons, mitochondria moved better. Right? They moved longer, they moved more persistently, presumably because myosin isn't breaking up microtubule track. They were also almost twice as long as normal. So they weren't so long that they didn't move, but they were really long. And we didn't get that with myosin 6 knockdown and myosin 2 knockdown. So there's a relationship between motility and fission fusion, but we don't really know what it is. If you have a fly that's mutated in DRP1, the primary defect is mitochondria don't get to the distal axon. If you have a fly that's mutated in mitofusin, they also don't get to the distal axon, just as if you mutated kinesin. So there's clearly a relationship between aspects of biogenesis and motility that's not made itself clear to us yet. Something to scratch our heads with. Other questions? Well, this is a terrible death to be talked about.
Okay, thank you. And we'll come back at 2 o'clock. Thanks a lot.
Oh, sure.
I have several questions, but I will discuss them. First, the idea is that someone should summarize the whole experience of this meeting. I know that you have not been here. I think that most experience so actually I had taken my I think that the next thing is yeah, it's just some visits. So it's kind of in the mix of somebody
Yeah, I'll think about it. And that's tomorrow or the we have a place where we see staff.
No, you were speaking
What we can't say is that what we can't visualize is specifically the hormones. We can't locally visualize it. One thing that we could do that we can in the sense that if we inhibit PI2 kinase a couple different ways pharmacologically, we can block the change in these well, no, we put the bead in and see that we get a change in potential. We put the bead in and put in the inhibitor. Yes. Right now, this is what didn't seem like the difference, although it did in the case of the location.
That means ESP is not just means linked to insulin receptor. What are you?
Well, it's linked to many receptors.
Most insulin is related to this PH.
Okay.
No, come with us. We can walk you.
Okay. I promised the student they wanted me to turn one of my presentations into a PDF that they could have.
This one's not.
Well, I'll just do all three. It takes a second. So let's go ahead.
Yes.
Yeah, I'll just.
And then that starts happening, you see enhanced and the model are also possible. Which is a paper from what if you go to government's website or the government research,
It is unbound and then we propose there is simulated evidence of clustering increases the velocity of.
I don't know what experiment. Let's not start another model. One is enough. Just as a movie. Be ready for it. It should not like suffer this. So the introduction we were okay, now we're going to the model. You're the model. Okay,
spatial and translational feeling. And the model is going to be strings attached to the target with a string constant and a relaxer and with a fixed temperature.
So this is a bad way to look at it, because it's just reading what is written on the reference. Okay, that's not how we should do it. If you're trying to learn, you have to say the same thing, but not just reading word by word. There's no point. Okay? Well.
And microtubules are assumed to be cylindrical structures with a specific diameter, and it is assumed that the mitochondria